Hello students, welcome to EPG Pathshala. I am Dr. Sudhita Gautam from Hansraj College, University of Delhi. Today we are going to talk on module Genome, Structure and Organization from paper Structural Organization of Genome. Learning or outcomes, basis of molecular genetics, genetic diversity among organisms is due to different genomes of the organism, components of prokaryotic genome, why prokaryotic genome differs from the eukaryotic genome, components of eukaryotic genome, expressed and unexpressed regions, how expression of the coded genes are regulated by the chromosomal elements. Introduction. The word prokaryote, it comes from Greek word pro meaning before and karyon meaning nucleus. Thus, the organisms which lack nucleus are referred to as the prokaryotic organisms. Genome is the entire DNA present in an organism's cell, that is all the genes. Genome data has helped us to understand the complete sequence of prokaryotic and eukaryotic genes. There has been an enormous increase in the genome data from 17 in 1998 to 149 in 2003. With the advancement of sequencing technology, with the rise in databases and galloping knowledge in the field of bioinformatics has helped us in understanding the enriched genome with good insights and better understanding. The chapter emphasizes on the organization of genome in prokaryotes and eukaryotes. Traditionally, it was believed that the prokaryotic genome consists of a single circular molecule of DNA. However, certain bacteria often have two sets of chromosomes, as in the case of Vibrio cholera, which has two circular chromosomes, one for the major housekeeping functions like that of growth and pathogenicity, and the second chromosome responsible for DNA maintenance, that is mainly involved in DNA damage response or repair mechanism. Prokaryotic genomes can vary a lot to extent of 100,000 base pairs or more. Example, Escherichia coli, Prochlorococcus marinus and Streptomyces coelicolar. 46, 56 and 75 is the size of the genome. In addition to the variation, even overlapping genome sizes are very common among prokaryotes. Beta proteobacteria and gamma proteobacteria. Chromosomal studies early in this century revealed that certain regions remained condensed during the interphase and prophase, while the other chromosomes were still as long and thin threads. These were called as heterochromosomes by the earlier cytologists and considered to be the sex chromosomes. However, Hates in 1929 showed that differential condensation was not limited to the sex chromosomes and was part of an otherwise normal chromosomes. Thus, two names, heterochromatin and euchromatin, were coined to describe the unusually condensed and typical segments, respectively. Chromatin is a feature of eukaryotic cells whose structure depends on several factors. Example, stages of the cell cycle. During interphase, the chromatin opens up and allows the SS of DNA and RNA polymerases that replicate and transcribe the DNA. The unwinding of the chromatin regions depends on the coding and non-coding regions of the DNA. The coding genes which need to be actively transcribed are loosely packed to facilitate the binding of RNA polymerase, whereas the DNA coding inactive genes remain associated with the structural proteins and are more tightly packed. Genetic diversity among organisms is due to the differences in the genomes. Prokaryotic genome is mostly haploid, circular, present in the cytoplasm and also has plasmids, which code the non-essential genes. Eukaryotic genome is larger in size, having a lot of non-coding regions and is usually present in diploid number, enclosed within a nucleus and marked mostly by the absence of a plasmid. The genome has a tendency to evolve with time through various processes like horizontal gene transfer, gene duplication, or lineage specific gene loss, etc. The evolution of the genome is beneficial to the organism in terms of better survival and helps to adapt or tide over the unfavorable period. Prokaryotic genome has been extensively studied by using E. coli as the model organism. The genome size of E. coli K12 is 4639 kb, which comes to about 2/5 the size of yeast genome, having 4405 genes. High level of coupling is seen in the various cellular processes in the prokaryotic genome. For example, transcription is coupled with translation because of absence of nucleus. Genes are expressed as operons present in chromosomes of relatively uniform chromosome sequence and gene density. Additional genes of the genome are present on separate circular molecules of DNA known as the plasmid. Differences between prokaryotic and eukaryotic genome. Prokaryotes contain a single circular chromosome confined in a nucleoid. Eukaryotes contain multiple linear chromosomes and the shape varies according to the cell stage. Prokaryotic chromosomes lack centromeres and telomeres. Eukaryotic chromosomes require centromeres, telomeres, and origin of replication to be maintained during cell division. Prokaryotic DNA can interact with the cytoplasm as nucleus is absent, thus transcription and translation can undergo simultaneously. Eukaryotic DNA is coiled around histone proteins to become compact and is enclosed within the nucleus. Prokaryotic chromosomes are haploid. Majority of eukaryotic cells are diploid. Prokaryotic transcription and translation are coupled as nucleus is absent. In eukaryotes, site of transcription and translation is nucleus and cytoplasm respectively. In prokaryotes, extrachromosomal plasmids are present and they provide desirable traits to the bacteria such as antibiotic resistance. In eukaryotes, plasmids are absent mostly. Prokaryotic genome do not contain a lot of repeated sequences, thus they are compact and efficient. Eukaryotic genomes have a large amount of repeated non-coding regions. The bacterial chromosome is tightly packed so that it can fit into the small volume of the bacterial cell. It involves supercoiling of DNA and further twisting the twisted chromosomes. Scaffold of RNA and protein contributes 50% to the chromosome and DNA domains are held together. The entire nucleoid is attached to the cell membrane. It helps in the segregation of the chromosomes during cell division. Histone proteins are absent in bacteria which are present in eukaryotes. However, it is believed that polyamines, organic molecules with multiple amine groups such as spermidine as well as some basic proteins are present in prokaryotes and help in reducing the size of bacterial chromosome. Chromosomes are circular in prokaryotes. Replication starts at single point called ori C and proceeds in both directions till the two replication forks meet. The chromosome is highly varied and ranges from a single circular to double circular or even a linear molecule of DNA. Example, Agrobacterium tumefaciens has two chromosomes, a linear chromosome of 2.1 MB and a circular chromosome of 3 MB. The chromosome can be easily distinguished from the plasmid due to its large size. E. coli has a supercoiled chromosome. The reason to which the DNA remains confined is known as the nucleoid. Changes in the genes are much more prone than the change in entire chromosome due to the continuous process of selection operating within the genes. Base composition patterns of the prokaryotic genomes reveal that there is a direct correlation between the genome size and gene content of the genome. GC content is higher in the genomes of soil-dwelling prokaryotes and gets reduced in genomes of host obligates and tend to have more of AT content. Free-living bacteria have shown to contain about 49% of GC content, and the figure is 38% in the case of host obligates. Another reason for this shift of nucleotides is due to the unchecked point mutations. Different regions of chromosome show different concentrations of A, T and GC nucleotides. It has been found that AT is in abundance in the intergenic non-coding sequences and the vowel position in coding sequences for reduced sized genomes. Upstream promoter regions are AT rich to facilitate a curved rigid conformation which unwinds more easily. The distribution of GC does not follow a set pattern in the genome. However, it has been observed that a preference of GC is near the origin and the AT is preferred near the origin of replication. The pattern of GC nucleotide distribution is also dependent on the whole host of functional and environmental circumstances. The biased distribution of G and C in an individual strand of the genome is known as the GC skew. GC skew is the measure of G minus C upon G plus C for leading and lagging strand in a bacterial genome. Plasmids are the extra chromosomal DNA in a prokaryotic cell capable of independent replication, stably inherited. The prokaryotic organism can survive even in the absence of a plasmid. The number and shape of the plasmid varies in different organisms. Human colon bacterium E. coli has been used extensively for the study of prokaryotic genome. Studies in 1950s and 60s made use of phase contrast microscopy and autoradiographic techniques to reveal the presence of a single circular chromosome having the essential genes of E. coli and packed within the cell nucleoid. The parameters which best convey bacterial genomes are the size, shape, replicon number, geometry, and the GC content. In prokaryotes, gene expression is transcribed with the help of operons. Earlier, the complexity of the genome was related to the genome size, now referred to as the C-value paradox. C-value paradox refers to the total amount of DNA in a haploid genome. According to the C-value paradox, there is a lack of correspondence between the C-value and organism, with the increasing level of complexity in the organisms. Plasmids are different from the chromosomes. They are found adjacent to the chromosome, smaller in size, having non-essential genes. Genes not involved to carry the housekeeping function. Instead, they give resistance from antibiotic and helps to grow in specific adverse conditions. Thus, they help in providing an additional selective advantage to the host. Example are factors. Additional beneficial traits include heavy metal resistance, degradation of complex molecules, disease-causing factors necessary for infection of plants or animal cells, which are passed on to the host bacterium through the process of conjugation. Three major types of bacterial plasmids have been extensively studied. F and F' plasmids, these are conjugation fertility factors. R plasmid, these plasmids have genes that confer resistance to antibiotics or other antimicrobial drugs. Col plasmids, these are the plasmids that have genes encoding coalescence proteins that kill sensitive E. coli cells. Other plasmids are also present that code for bacteriocins and vibrocins. Vibrocins have the capability to kill sensitive Vibrio cholerae. Plasmids may be divided into two groups, conjugative or transmissible plasmids. These plasmids mediate the transfer of DNA by conjugation. Examples F and F' plasmid. Many plasmids and some col plasmids. Non-conjugative or non-transmissible plasmids. These plasmids do not mediate transfer of DNA by conjugation. However, Fraser et al. In 1997 has reported the presence of more than 20 circular and linear plasmids in Borrelia, which code for genes involved in infecting ticks and humans. They have an inbuilt capacity of integrating into chromosome and pass from one cell to another, often of small size, that is, less than 1500 kilobases, and bearing innate capability of self-replicating. Plasmids have been extensively used by molecular biologists as a cloning vector in laboratories to transfer external DNA into a cell. Up to 10 kb of DNA has been engineered and used for gene manipulation. Techniques like gel electrophoresis or cesium chloride buoyant density gradient centrifugation can be used to easily identify plasmids from chromosomal DNA. Example, Bacillus thuringiensis has a circular chromosome 5.7 MB and 6 plasmids, and each plasmid is more than 50 MB. Eukaryotic genome has two sets of chromosomes, one from each parent. It differs from the prokaryotic genome in bulkiness as it has a lot of non-coding DNA, also referred to as the junk DNA. Junk DNA acts as a sink to absorb a lot of mutations. The entire eukaryotic DNA is confined in the nucleus and remains associated with proteins. The association varies depending upon the requirements of the cell at various stages of the cell division. It's a multi-level system of DNA packing. Chromatin is formed when the double-stranded DNA is wound around histone proteins to pack and condense the bulky DNA. The word chromatin was first used by Walther Flemming. Flemming explored cytology, that is, the study of chromosomes, and studied the process of mitosis in detail. He even revealed the movements of chromosomes. In 1979, he stained salamander embryos using aniline dyes, which are byproduct of coal tar, and was able to identify and visualize the chromosome movements during cell division, which he described in his book Zell Substance Kern and Zellteilung, published in 1882. Episomes are distinguished from other extrachromosomal DNA like plasmids on the basis of their size. Usually they have a molecular weight of 62 kb. It is a form of plasmid which can replicate on its own in addition to the ability of integrating into a bacterial chromosome. It does not form part of the genome and is a non-essential element which originates outside the host. It exists in various forms. First, F plus form as an autonomous extrachromosomal form, and HFR form, high frequency recombination form. They are passed on to the daughter cells as they multiply with the host cell division. Both the forms, that is F plus and HFR forms, are capable to donate genetic material during conjugation in various bacteria, example Salmonella, E. coli and Serratia. Conjugation results in converting a F negative cell lacking episome into an F positive cell or an HFR cell, depending upon the conjugating partner type. Another type, known as F or F prime, found outside the chromosome but with a pair of chromosomal DNA attached to it. Chromatin is present almost in all cells with an exception of erythrocytes. During interphase, chromatin is distinguishable as two different forms, that is, euchromatin and heterochromatin. Histones are the most abundant proteins in chromatin. They are mostly made of lysine and arginine, amino acids, which are responsible for contributing a net positive charge to histone proteins. Positive charge is helpful in binding of the negative charge of the phosphate molecule in DNA. This complementarily of the charges helps in holding the DNA in close contact with the histones. 5 types of histone proteins associated with the chromatins are H1, H2A, H2B, H3 and H4. Changes in the chromatin organization helps in the process of replication and transcription. During the process of replication, histones leave the DNA only transiently, whereas they stay with the DNA during transcription. The changing shape and positional variation of the histones provide the movement of the RNA polymerase along the DNA molecule. Core histones consists of H2A, H2B, H3 and H4. There are 2 molecules of each type of core histone proteins, which results in the formation of a histone octamer complex. H1 is referred to as a linker histone, which seals the entry or exit position of the DNA strand on the nucleosome. H1 histone and the nucleosome bead together is known as chromatosome. Binding of the DNA and nucleosomes is highly non-specific depending upon the requirement of the cell and their function in general DNA packing. Chromatin structural organization. consists of a multi-level system of packing the DNA. Histones are the first level to initiate DNA packing. Uncoiled chromatin appears like a string with the beads of nucleosomes. In 1974, the first level of chromatin folding was described by Roger Kornberg. Chromatin is a feature of eukaryotic cells whose structure depends on several factors. Example, stage of the cell cycle. During interphase, the chromatin opens up to allow the SS of DNA and RNA polymerases that replicate and transcribe the DNA. Functions of chromatin: packing of DNA to fit within the cell's nucleus, to allow flexibility to DNA for carrying out the processes of mitosis, to protect DNA from damage, and to regulate and control the genetic expression through DNA replication and transcription. Chromatin is easily distinguishable into two phases during interphase: euchromatin, which consists of active DNA, that is, the region which code for specific gene and is being expressed as protein and repetitive sequences are absent. Second, heterochromatin, which consists of mostly inactive DNA or the DNA which is not expressed as genes or proteins. Functional chromosomal elements consist of promoter sequences, terminator sequences, enhancers and silencers whose position varies in prokaryotes and eukaryotes. Promoters are DNA sequences located at the 5' end above the transcription, initiation or the start site. Termination sequences are sequences of DNA just downstream the gene coding region recognized by RNA polymerase and which results in the termination of the transcription process. Placement of enhancer or silencer can be ahead of insulator and the initiator sequence. Enhancers are a DNA sequence which are regulatory in nature but capable of enhancing the transcription of a gene. Silencers are the sequence of DNA responsible for reducing or shutting off the expression of a gene. Insulators are a sequence of DNA responsible for regulating the effect of silencers or enhancers. So students, let us now summarize what we have learned in this module. Genome is the entire DNA present in an organism cell, that is, all the genes. Prokaryotic genome is mostly haploid, circular, present in the cytoplasm and also has plasmids which code the non-essential genes. Eukaryotic genome is larger in size, having a lot of non-coding regions and is usually present in diploid number, enclosed within a nucleus and marked mostly by the absence of a plasmid, sometimes present in eukaryotes. The genome has a tendency to evolve with time through various processes like horizontal gene transfer, gene duplication or lineage specific gene loss, etc. The evolution of the genome is beneficial to the organism in terms of better survival and it helps to adapt or tide over the unfavorable period. The bacterial chromosome is tightly packed so that it can fit into small volume of the bacterial cell. It involves supercoiling of DNA and further twisting the twisted chromosomes. Chromosomes are circular in prokaryotes. Replication starts at a single point called ORI-C and proceeds in both directions till the two replication forks they meet. The parameters which best convey bacterial chromosomes are the shape, size, replicon number, geometry, and the GC content. Plasmids are the extra chromosomal DNA in a prokaryotic cell capable of independent replication, stably inherited. The prokaryotic organisms can survive even in the absence of a plasmid. Episomes are distinguished from other extra chromosomal DNA like plasmids on the basis of their size, and usually they have a molecular weight of 62 kb. It is a form of plasmid which can replicate on its own in addition to its ability of integrating into the bacterial chromosome. Eukaryotic genome has two sets of chromosome, one from each parent. Chromatin is present almost in all cells with an exception of erythrocytes. During interphase, chromatin is distinguishable into two different forms, that is Euchromatin and Heterochromatin. Nucleosome forms the basic unit of chromatin joined together by short stretches of DNA itself. Changes in the chromatin organization helps in the process of replication and transcription. Functional chromosomal elements consists of enhancers, silencers, insulators, promoters, and terminators, which help in regulating the gene expression. Thank you.